Our protocol describes a method to build an RA model. Its success rate is over 93%, and its pathogenesis depends on T cells, B cells, and innate immunity, which allows researchers to better study the pathogenesis of RA from a global immune perspective. Then we use iNKT cells with immunomodulatory effect for their treatment. iNKT cells have a good effect after treatment by in vivo introduction, which provide basic data support for the clinical application of iNKT cells, and fully exploits the clinical application value of iNKT cells. The greatest advantage of this technology is that it is simple to operate and easy to copy. Demonstrating the procedure will be Chen, Shengde, Xiang, and Zhang Jingnan, the graduate students from my library.
To begin, weigh 1.75 milligrams of both HGPI 325-339 and HGPI 469-483 fragments and dissolve them in 5.25 milliliters of 4 degrees Celsius triple distilled water. Dissolve complete Franz adjuvant in a 50 degrees Celsius water bath. Draw 5.25 milliliters into another 10 milliliter centrifuge tube and cool it for use. Put the mixture of G6PI solution and complete Franz adjuvant solution in an artificial emulsification unit with 2 glass syringes connected. In an